All right. Hey, everybody. Let's practice one of these transcription translation sort of like run-throughs, essentially. I wouldn't say that this is how every transcription translation problem is going to go, but there is a semi-robotic way to solve these for everything you need. So let's take a look right here at the pencil rolling. Here's a strand of DNA noting the directions, and I'm asking, show me all the way, what are the tRNA anticodons that are going to bind this? Along the way, we're going to have to make the mRNA. We may as well make the proteins, and we may as well eventually go all the way back in time and see the coding DNA of this one. So, and note, whenever you're reading these, physically transcribed to make the mRNA, we consider this the template strand. Always kind of read and note directions of stuff, because otherwise things can get a little messy. So I'm going to go ahead and let's go black here. I'm just going to copy it down here for myself as we go. Here, I'll even go purple here. So three primes. We got T, A, C, G, T, A. There we go. Okay. And I'm going to say this is the template DNA. There we go. So the template DNA is going to be the one that copies and is copied by the mRNA piece for piece. So if I transition to black right here for the mRNA, no matter what, my mRNA is always going to have to go the opposite direction. So that's why the directions have changed now. So T on the DNA will be an A to the mRNA. An A on the DNA, though, is going to be a U to the mRNA. And then C's and G's are just the same, okay? So T and A and C. So just those two little uracils are going to be the changes. We shift over here into RNA. And even we're just going to say mRNA in this case. We're not going to do any splicing here. Okay, so we're here. We may as well go take a look quickly at what these proteins are. So AUG, CAU, let's take a look. Oh no, I'm going to have to scroll. I'm sorry. CAU is histidine and AUG is our methionine. Not bad. Okay. So you would have the table on there each time. So that's when I would say you should be okay. We're just going to go ahead and put methionine, histidine right there. Next, I'm going to change colors here. Let's actually investigate the tRNA that is actually binding each one. And again, directions are going to have to change each time, because you always have to run anti-parallel, kind of like a two-way highway, essentially, right? So now that we've got this, you can still just make the easy transition down to each one for RNA. U, A, C, just like that. C's go to G's, A's still go to U's, and U's still go to A. So there we go. I think we have it right here, 3' to 5'. GUA, GTA, or sorry, whoa, whoa, whoa. Give me that back. Gotta be careful. But looks good. There's your answer right there. If you want to go another level, I'm just going to say coding DNA, because remember, coding DNA always has two helices, so you can just check out the other one right here. Notice how the coding DNA, again, you have to change directions even when you're doing double helix like this. Coding DNA is going to look like the mRNA. That's why we call it that. It is not the one actually being transcribed, though. That's the more important one, the transcription or the template DNA. That's the key. So hopefully this little mini video helps here. Maybe I'll make a fun like speed video out of it eventually.